A very good afternoon to all. So today we'll be starting with locomotion and movement. I'm launching the poll. Quickly answer through poll. My audio is clear and my screen is visible. Hello students, how are you all? Quickly answer through poll. My screen is visible and my audio is clear. Okay, so today we'll be discussing locomotion and movement. Okay, as you know that there's a difference between movement and locomotion. Movement when there is no change in place or location. For example, I can move my forelimbs, we can move our eyelids, like jaw movement. These all are movement because there is no change in the place or location. But when there is a change in place and location, we call it as locomotion. And these movements are voluntary in nature. Voluntary means that they can be controlled, that they are under our control, and it can be controlled through the nervous system, like walking, running, climbing, flying, swimming. All these are locomotory movements because there's a displacement is taking place in these movements. There's a change in place or there's a change in location. If there is a change in location and place, we call it as locomotion. Okay, and these movements are voluntary. There are types of movement. In cells of human body exhibit three types of movement. One is amoeboid, ciliary, or muscular movement. Amoeboid is shown by macrophages or leukocytes that neutrophils, phagocytosis. Right, the engulfing of the foreign particles, which are shown by macrophages or the monocytes or neutrophils of the leukocyte. They show phagocytosis, which is one of the feature of amoeba. That is why they are calling it amoeboid movement. Now there's a ciliary movement, which is most of our internal tubular organs, which are lined by the ciliated epithelium. They show the ciliary movement. And third is the muscular movement, which is shown by our limbs, jaw. Okay. Muscles, as you know, that muscle has a mesodermal origin. Remember the origin. It is mesodermal in origin. It is a specialized tissue, connective tissue. And it constitutes 40 to 50% of the body weight in human adult. And there are special properties of muscles like excitability, contractibility, extensibility, and elasticity. First, the muscles that they are of three types. One is skeletal muscle, there's cardiac muscle, and the visceral muscles. Please remember the feature of these muscles because they are very important. Skeletal muscles, they are closely associated with the skeletal component of the body. They are also known as striated muscles. They're also known as striated muscles because they show striation. That is, they show light and dark band alteration. You can see these. There's a light and dark band. There's an alteration is there. because of which they are known as a striated muscles. And these muscles, they are voluntary. They are voluntary in nature. That means they are under our control and they can be controlled by the central nervous system. These muscles, they are cylindrical in nature. You can see they're cylindrical like this. They're cylindrical. They are unbranched. Remember these points. Next comes the visceral muscle. Visceral muscles, visceral means internal organs. So the muscles which are forming the internal organs are the visceral, and these are involuntary in nature. Involuntary means that they are not under our control, or they are not controlled by the nervous system. Their activities are not under the voluntary control of the nervous system. Therefore, they are known as the involuntary muscles. They're also known as smooth muscles. Visceral muscles are fusiform in shape. Remember this, like this, they are fusiform. And they're tapering at the ends. Their ends are tapering, pointed ends. In the center, there's a nucleus is present, so they are uninucleated. Skeletal muscle, they are multinucleated because they consist of muscle fibers, and muscle fibers, they're also known as muscle cells, and they're multinucleated. That I will tell you later when we will discuss the structure of the skeletal muscle. So here, the visceral muscles, they are fusiform in shape, they are uninucleated, and they're also unbranched. One more important point, that they're non-striated muscles. They does not show striations. There's no light or the dark band alteration. And they're mostly found in the lining of the digestive tract, genital tract. Next comes the cardiac muscles. As the name suggests, cardiac. Cardiac means heart. So these muscles, they are found in the heart. Cardiac muscles, they are structurally like the voluntary muscles, like skeletal muscles, but they function as involuntary muscles. So these are involuntary in nature. Cardiac muscles are branched. Please remember this point. Cardiac muscles are branched, whereas the skeletal muscles and the visceral muscles, they are unbranched. But cardiac muscles are branched like this. Okay, there you can see this branching. They are branched. And they also have a presence of intercalated disc. Very, very important point. Please remember this. And the function of this intercalated disc is in the transmission of impulse. Quick transmission of impulse. Okay, that impulse can be transferred from this muscles to this, from this to this. So they help in the quick transmission of impulse, and they are the intercalated disc which are present in the cardiac muscles. And these muscles, they are striated, but they're faint. They show faint striations. Moving next is the structure of the skeletal muscle. Skeletal muscles, please remember this, that the functional unit of skeletal muscle is sarcomere. And their anatomical unit is muscle fiber. They are important point. Please remember this. And the skeletal muscles, they consist of bundle of muscle fibers, and these bundle of muscle fibers are known as fascicles. You can see these bundles, they are the fascicles. They are the bundles of the muscle fibers. And these bundles, they are held together by the common collagenous connective tissue, which is known as fascia. Here, these are the collagen fibers, which are holding these fascicles together. If I take out one fascicle, the fascicle, they consist of muscle fiber because they are the bundle of muscle fiber, and the muscle fiber, they are also known as muscle cells. And these muscle fiber, they are lined by a membrane which is known as sarcolemma. So this is a sarcolemma. On the sarcolemma, there's many nucleus, multinucleated. So the muscle fiber, the muscle cells, they are multinucleated, and the nucleus, they are present towards the periphery. These nucleus are present towards the periphery. And in the middle, there's a fluid-like structure. Fluid is present, which is known as sarcoplasm. And in this sarcoplasm, there are microfibrils are present. These are myofibrils. Myofibrils are present, and myofibrils are consist of actin and myosin. Which form the light band or the dark band. The myosin will form the dark band or the A band, and the actin will form the I band, which is a thin band or the light band. We also know that these muscle cells, they also consist of sarcoplasmic reticulum. Sarco means muscles. Remember this term. Sarco means muscle. So there's a endoplasmic reticulum which are present in muscles. They are known as sarcoplasmic reticulum, and these are the storehouse of the calcium. Here the calcium is stored in the sarcoplasmic reticulum, and these calcium, they play a very important role in contraction of the muscle. And the myofibrils, they consist of two filaments, which are actin and myosin. Myosin are the thick filament, and actin are the thin filament. Actin filament, they form the I band, which is also known as isotrophic band. And the myosin, they form the A band, which is known as an isotrophic band. And these bands, they show alteration. Like first there will be I band, then A band, then again I band, then A band, then I, then I. So they show alteration. Now let's discuss the structure of the sarcomere. As you know that the functional unit of the muscle fiber of the skeletal muscle is sarcomere. So these red line, they are the actin filament or the thin filament, right? And there's a line which is known as a Z line. This is a Z line, the green line. These Z line, they are bisecting or dividing the actin filament. You can see that these Z line or the green lines, they are bisecting the actin filament. And the portion which is present between the two successive Z line is known as the sarcomere. These are the sarcomere. From here till here is the I band. And I band only consists of actin filament or the thin filament. Remember this. Similarly, from here till here is the I band, and I band only consists of actin filament. And these actin filament, they are held together or they are firmly attached with the Z line. In the middle, there's a blue filaments are present. These are the thick filament or the myosin filament. From here, the length of the myosin filament represent the A band. Remember, the length of the myosin filament represent the A band. So A band will be from here till here will be the A band. And in the middle, there's a M line is present. This is a M line. In the middle of the thick filament, there's a M line are present, which are holding these thick filament in the center. Okay. And there's a portion is present where only the myosin filament are present and the actin filament are not there. This portion is known as a H zone or the Hansen zone. This portion, because here only the thick filament or the myosin filament are present and the thin filaments are not present. So this zone is known as H zone. In the resting stage, when the muscle are in relaxed stage, in that case, the actin filaments, the edges of the actin filament, they partially overlaps the free ends of the thick filament or the myosin filaments. And the portion, this is the H zone. The portion of the thick filament which is exposed, this is known as the H zone. From here till here is the I band. From here till here is the A band. And again the I band. So you can see the I band, then A band, then I band. So there's an alteration of band. But during the contraction, these thin filament, they start overlapping on the thick filament. They start overlapping, and this H zone, which is present in the relaxed state, when the muscle is in the relaxed state, this H zone starts reducing and finally disappear when the muscle is fully contract, because these thin filament, they start overlapping, or they start sliding over the thick filament. Okay, these thin filament, they start sliding over the thick filament, and it leads to the contraction of the muscle. So please remember that the length of the myosin or the thick filament remain as it is. It is the length of the I filament which is reducing. Okay, length of the A filament or the length of the myosin will remain as it is, and the length of the I filament or the actin filament, they start decreasing because they are pulling inward. Next are the contractile protein. So there are two types of contractile protein, which are actin and myosin. Actin are formed, first is the G-actin. This G-actin or the globular protein, this is a globular protein, like this. This G-actin will undergo polymerization and it will form F-actin. Now two F-actin were helically coiled and they form one actin filament. And these actin and one more important point, there are two regulating proteins. One is tropomyosin and the another one is troponin. Two filament of the tropomyosin, they run closely with the actin filament. These orange color filaments, these are tropomyosin, and they are running closely with the actin filament. Function of the tropomyosin is to prevent the interaction between the actin filament and the myosin filament. They're preventing the interaction between the actin filament and the myosin filament. Another protein is the troponin, and troponin is a complex protein. Why it is complex? Because it contains 3 units. There is IPC, which is a calcium binding protein. Second is TPT, which is a tropomyosin binding protein. And the third one is TPI, which has inhibition, role in inhibition. TPI, what What this protein does, it covers it masks the myosin binding site of the actin and does not allow the interaction between the actin and the myosin. Moving next is myosin. Myosin is also formed by the polymerization of a unit which is meromyosin. Meromyosin is consists of two parts. There's heavy meromyosin and there's light meromyosin. Heavy meromyosin is consist of globular head and a short arm. And light meromyosin is consist of tail. Head is further consist of two binding site. You can see there two binding site. This is a head. And the two binding sites are the actin binding site and the ATP binding site. Now let's come to the mechanism of muscle contraction, very, very important. And it is also known as the sliding filament theory because the thin filament, they are sliding over the thick filament. That is why we call it as sliding filament theory. First thing which happened during the mechanism of muscle contraction is the signal. The signal are sent by the CNS or the central nervous system by a motor neuron. Let's discuss with the help of this image. First is the signal sent by the central nervous system through the motor neuron. This motor neuron will reach or come in contact with the muscle fiber or the muscle fiber cells. The junction which is formed is known as a neuromuscular junction. Now this motor neuron and the muscle fiber, they will act as a motor unit. When the signal reaches the end of the motor neuron, these vesicles which are containing the acetylcholine, which is the neurotransmitter, they will release into the neuromuscular junction. You can see this red. They are the acetylcholine neurotransmitter which are released in the neuromuscular junction. Once they get released, the receptor which are present in the sarcolemma, which is the lining of the muscle fiber or the muscle cell. The receptor which are present in the sarcolemma, they receive this neurotransmitter and generate an impulse. This impulse will be transmitted to the T tubules of the sarcolemma. And from the T tubules, these impulses, they are given to the sarcoplasmic reticulum. Once the impulse reaches to the sarcoplasmic reticulum, the calcium channels which are present in the sarcoplasmic reticulum, they get open and all the calcium, they comes out in the sarcoplasm. Now all the calcium, they start moving into the sarcoplasm and they bind to the calcium now bind with the TPC, which is a troponin calcium binding protein. They will bind to the calcium. Once TPC binds with the calcium, there's a conformation changes occur in TPC. And because of the conformation change, the TPI, remember, which is inhibiting the interaction between the actin and the myosin, now it's shift, it slides a bit or it slides down, because of which the tropomyosin also get loosened and the myosin binding site on actin get exposed. So till this end, now the myosin binding site on actin get exposed. At the same time, there's an event is also taking place in the myosin, which is the ATP hydrolysis. This event is taking place in myosin filament, which is thick filament. As you know, the myosin head has two binding sites. One is the ATP binding site and the actin binding site. So the ATP will bind to the ATP binding site, and you know that the globular head of the myosin, it has the ATPase activity. So this ATP will be broken down to ADP and PI. Okay, so when this ADP is released, the head of the myosin get binds, like this is a thin filament, okay, actin filament, and this is the head of the myosin. So when the ADP release, this myosin will get attached to the actin filament, okay. And when the PI released, when this inorganic phosphate released, in that case the head of the myosin will pull the actin filament inward. This is how the sliding movement takes place. First, this is an actin filament. When the myosin head binds to the actin filament, when this ADP is released out, and when the inorganic phosphate release, this actin filament will move like this. It will pull inward with the help of this myosin head. Myosin head will pull the actin filament inward. And during the relaxation state, now the contraction has done. This is how the contraction takes place, because now these myosin head, which are stick or the binds to the actin filament, now they will pull the actin filament inward and the contraction takes place. Now during the relaxation, what happened? Now this ADP, which is released in the muscle, there's a protein is present which is known as a phosphokeratin. These phosphokeratin protein, they will donate their phosphate to the ADP, and ADP will be converted to ATP. Okay, now the ATP has regenerated. Now this ATP will move and bind to the myosin head again, which is attached to the actin filament. Once this ATP bind again to the myosin head, now the myosin head will get detached from the actin filament. Second point which takes place during relaxation is the acetylcholine. We know that these neurotransmitters, they generate impulse. They have a role in generating impulse. Now, during the relaxation, we have to remove these neurotransmitter or acetylcholine. So there's an enzyme is released, which is known as acetylcholine esterase. These enzyme will digest the acetylcholine. Okay. And now we know that during the relaxation, we have to influx all the calcium which are released during the when the impulse is received by the sarcoplasmic reticulum. Now, during the relaxation, we have to influx all the calcium back to the sarcoplasmic reticulum. So in this process, there's also a protein is required, and this protein is known as calciquestin. This is the protein which influx the calcium back into the sarcoplasmic reticulum during the relaxation state. Now I have discussed both the contraction and the relaxation. Now you know that muscle fibers, they are of two types. There are red muscle fiber and there's a white muscle fiber. Red muscle fiber are those which have a very high amount of myoglobin. And you know that myoglobin, they are the storehouse of the oxygen. So they have more myoglobin, because of which they give the reddish appearance, and we call it as red muscle fiber. They also have a large amount of mitochondria. Remember this, the red muscle fiber, they have large amount of mitochondria, that will more, and they have more myoglobin. So if there is more myoglobin, there will be more oxygen. So mitochondria will perform the aerobic respiration and it will produce a large amount of ATP. So the muscles which are having this red muscle fiber, they does not show fatigue easily, like our extensive muscles, back muscles, our extensive back muscles, they does not fatigue very easily. Now, second is the white muscle fiber. They have a less myoglobin, because of which they give a white or the pale color. They have less mitochondria. And these muscles, they show rapid contraction, because of which they require more calcium. And if they require more calcium, that means they will have more sarcoplasmic reticulum. Whereas the red muscle fibers, they show slow and continuous contraction. So they require low calcium. And white muscle fibers, because they have less mitochondria and they have less myoglobin, that means they would have less oxygen. So there's a chances that anaerobic respiration takes place in white muscle fibers, because of which we know that when the glucose is broken down to pyruvate, when there is insufficient amount of oxygen or when there's a depletion of oxygen, this pyruvate undergo fermentation and produce lactic acid. And we start feeling fatigue. Now this lactic acid has two fate. This 1/4 of the lactic acid will be oxidized and produce carbon dioxide and water. 4/5 of this lactic acid will go to the liver, and liver will convert this lactic acid to glucose and again back to muscle. This cycle, which takes place in liver where the lactic acid is converted to glucose, we call it as Cori cycle. Okay. Now moving next is the skeletal system. Skeletal system is consist of two parts, which is bone and cartilage. Bone, they are hard and inflexible. They have deposition of salt, mainly the carbonate and phosphate, especially the calcium and magnesium salt. And the most abundant salt which is present in bone is a calcium phosphate. Please remember this, is calcium phosphate. Cartilage, they are soft, they are flexible, they have rare deposition of mineral salt. Now, bones are further divided into two systems. One is the axial skeletal system. Axial means which form the central axis of the body. The axial system is the one which forms the central axis of body, and appendicular skeletal system are those which form the appendages, like forelimbs or the hind limbs, plus the girdle. Girdle are the bones which attach the forelimbs and the hind limbs to the central axis of the body. Appendages skeleton, they consist of 120 bones, and this axial skeletal system, they consist of 80 bones. So total there are 206 bones. Now let's discuss the axial skeletal system. As the name suggests, axial, they form the central axis of the body. You can see they're forming the central axis of the body. They consist of skull, they consist of vertebral column, they consist of sternum, ribs, which constitute the axial skeletal system. Now let's discuss these one by one. Number one is the skull, which consists of the cranial bones and the facial bones. Cranial bones are 8 in number and the facial are 14 in number. So total there are 22 bones. Cranial bones, they consist of frontal bone, which protects the front part of the head. Like here is the frontal bone. Next is the parietal bone, which protects the roof of the brain. Third is the occipital bone, which protects the hind portion of the brain. Fourth is the temporal bone, which protects the lateral portion of the brain. There are sphenoid bone and the ethmoid bone. So these are the 8 bones which are present in the skull. Remember one point, there is occipital bone. They have two condyle. Like this. If this is an occipital bone, it has, these are called condyles. And because of which they are attached to the first vertebra, which is the atlas. So we mammals and amphibians, we have a dicondylic skull, whereas the birds, fishes, and reptiles, they have a monocondylic skull. Sphenoid bone, they have a depression because of the pituitary gland, and this depression or the cavity is known as sella turcica. Occipital bone also have a hole through which the brain exits as the spinal cord, and this hole is known as the foramen magnum. Now let's come to the facial bone. There's nasal bone, there's a lacrimal bone, inferior nasal concha, maxilla, which form the upper jaw, mandible, which form the lower jaw, there's a palatine, there's zygomatic, and there's vomer. So these are the 14 facial bones. And apart from this, there's also a hyoid bone. Hyoid bone is a U-shaped bone, and this bone is single. It is a single bone, and it does not form the joint with the other bone. And this bone, it provides the surface for the attachment of tongue muscles. They are present at the surface of the buccal cavity. They provide the surface for the attachment of the tongue muscles. Moving next is the ear ossicle. Ear ossicles, these are found in the middle part of the ear. These ossicles, they consist of three bones. There's malleus, incus, and stapes. Malleus is derived from the articulate bone, incus is derived from the quadrate bone, and stape is derived from the hyalomandibular bone. And the main function of this ear ossicle is the amplification of the sound waves. Now let's come to the vertebral column, which consists of 26 bones. They are further divided into cervical, thorax, lumbar, which means they are present in the upper abdominal, sacrum, which are present in the lower abdomen, and coccyx. Main function of the vertebral column is to support the head and protect the spinal cord. And there's a hollow canal is present, which is a neural canal through which the spinal cord passes. Most important is the sternum and rib. You can see this red color bone in the center. This is the sternum. It is present ventrally and it is a dagger shape. Dagger means khanjar shape. And the ribs are present. There are 12 pair ribs. The 1 to 7 are known as the true ribs. They are known as true ribs because they are dorsally, they are attached to the vertebral column, and ventrally they are attached to the sternum. Now, 8, 9, and 10, these are known as false rib. Because dorsally they are attached to the vertebral column, but ventrally they are attached to the 7th rib, which is attached to the sternum. Okay, so they are not directly attached to the sternum. First, they are attached to the 7th rib, and then the 7th rib is attached to the sternum. So they are known as a false rib. They are also known as vertebrochondral ribs. 11th and 12th, they are the floating ribs. Because on the dorsally they are attached to the vertebral column, but ventrally they are not attached. So these are known as a floating ribs. And thorax vertebrae, ribs, and the sternum together, they form the rib cage. Moving next is the appendicular system, which consists of forelimbs, hind limbs, and the girdles. each limb or the forelimb is consists of 30 bones. So if I multiply by 2, that means it will be 60 bones. This is the upper part of the arm. This is the lower part of the arm. Upper is consist of humerus bone, radicus. Lower is consist of radius and ulna. Wrist bone consists of carpals. Bones which form the wrist are the carpals. Then there are metacarpals which forms the palm, and there are phalanges which are present in our fingers. And this is the clavicle and scapula. They are the shoulder girdle. They are part of shoulder girdle. Moving next is the hind limb. Hind limb is consist of femur, which is the thigh bone. This is the strongest, longest, and the thickest bone. Here's the kneecap is present, which is known as patella. This lower leg is consist of fibula, tibia. The tarsals are present, the metatarsals are present, the phalanges are present. Tarsals consist, they form the ankle. Next, move to the girdle. Girdle, they are of two. There's a shoulder girdle and there's a pelvic girdle or the hip girdle. Shoulder girdle will join the forelimbs to the central axis of the body, and pelvic girdle will join the hind limbs to the central axis of the body. So shoulder girdle is consist of shoulder blade, which is known as scapula. And scapula, it divides into three parts, which is body, spine, and acromenon. Acromenon is attached to the clavicle, which is the collarbone, and clavicle is attached to the sternum. In this scapula, there's a cavity is present or a depression is present, which is known as a glenoid cavity. And this cavity, in this cavity, the head of the humerus bone get attached like this. This is a cavity, glenoid cavity, and this is a humerus head. They get attached or get fixed. Moving next is the pelvic girdle, or we call it as hip girdle. It consists of coxal bone, two coxal bone. Each coxal bone is divided into ilium, ischium, and pubis. There's a hole is present in the ischium, which is known as the obturator foramen. And there's a cavity is also present, or the depression is present in the ischium, which is known as acetabulum, where the femur bone attached, head of the femur bone attached. In the center, there's a fibrous cartilage is present, which is known as pubis symphysis. Now let's come to the joint. We know the joint are the point between the two bones or between the bones and the cartilage. It could be a fibrous joint, cartilaginous joint, or synovial joint. Fibrous joint, they are fixed joint. That means they does not show movement. For example, our skull bones, they are joint fixed. This is a, for example, this is a bone number 1, this is 2, and they're joined with the dense fibrous connective tissue. And this joint is fixed. It cannot move. Next comes the cartilaginous joint. In the cartilaginous joint, the two bones, they are joined by the fibrous cartilage. They're joined by the fibrous cartilage and they show slight movement. Synovial joint, for example, this is bone number 1, this is bone number 2. Both the bones, they are articulated. They're covered with the cartilage. There's a cartilage, which is the, and there's a synovial membrane. Over this, there's a synovial membrane. And this synovial membrane secrete the synovial fluid in the cavity, which makes the movement easy. So synovial joints, they show movement. They show slight movement, cartilaginous joint. And fibrous, they are fixed, they show no movement. Synovial joint are of further types like a hinge joint, condylar joint, ball and socket joint, saddle joint, pivot joint, and plane joint. Moving next is the disorder. First disorder is the myasthenia gravis. This is an autoimmune disorder, and because of this disorder, it leads to fatigue, weakening, and the paralysis of the skeletal muscles. Second one is a muscular dystrophy. It is a genetic disorder which leads to the degeneration of the skeletal muscle. Third is the tetany, which is a rapid spasms and which occur due to the low calcium level in the body fluid. Fourth one is arthritis, that is the inflammation of the joint. And remember, gout is the inflammation of joint, but this inflammation is due to the accumulation of the uric acid in the joint. Next is the osteoporosis. Osteoporosis is a weakening of bones and it occurs with the ages. Okay, is an age-related disorder. Because of as the age increases, the bone weakening also increases and the chances of fractures also increases. These are important. Please remember this. Now let's move to the question. Question number 1 says, which of the following statement wrongly represent the nature of the smooth muscle? They're asking about the smooth muscles. Option 1 says these muscles have no striation. Option 2 says they are involuntary muscles. Option 3 says that the communication among the cell is performed by intercalated disc. Option 4 says these muscles are present in the wall of blood vessels. What do you think? What will be the correct option for this question? They're asking the wrongly representing. They're asking the wrong statement with respect to the smooth muscle. Quickly answer. What do you think? What will be the correct option for this question? Very good, Chechni. Very good, beta. Smooth muscles, these are involuntary muscles. These muscles, they show no striation. These muscles are known as visceral muscles. These muscles are fusiform shaped. They are uninucleated, like this. The nucleus is present in the center. They are tapering at the ends. These are the smooth muscles. And the question is saying which of the following statement is wrong about the smooth muscle? These muscles have no striation. Yes, it is true. These are involuntary muscles. Yes, it is true. Communication among the cell is performed by intercalated disc. No, this is wrong. Because intercalated disc is a function of or is a feature of cardiac muscles. Remember the cardiac muscles, they are branched. And here the intercalated discs are present. And these intercalated discs, they help in a quick transmission of the impulse. These muscles are present in the wall of blood vessels. Yes, it is also true. So the communication among the cell is performed by intercalated disc. This is a correct option with respect to question because question is asking the wrong statement. Moving next. Calcium is important in skeletal muscle contraction because it binds to the troponin to remove the masking active site on actin for myosin, activates the myosin ATPase by binding to it. Third, detach the myosin head from the actin filament. Fourth, prevent the formation of the bond between the myosin cross bridge and the actin filament. What do you think? What will be the correct option for this question? They're asking the calcium, role of calcium in the contraction of the skeletal muscle. What do you think? What will be the correct option for this question? Quickly answer, beta, so that we can cover more questions. Okay. They are saying the calcium is important in skeletal muscle contraction. Yes, calcium is very important. Remember the TP, which is a troponin protein. And this protein is a complex protein. It has 3 units, TPC, TPT, and TPI. Very good, very good. So it has this troponin protein, it has 3 units. There's TPC, which is a calcium binding protein. There's a TPT, which is a troponin binding, and I is the inhibition, which is the role of this I is to mask the myosin binding site on actin. For example, this is the actin. Just imagine this is the actin. And here is the myosin binding site, the site where the myosin head will bind. Okay. And this is a tropomyosin. And here is troponin. This troponin has first it will 3 units. This TPT, which is a troposin binding, it is through which the troponin is bind to the tropomyosin. It has this calcium and this I. This is C, this is T, and this is I. You can see this I, it is masking or covering the myosin binding site on actin, because of which the myosin head cannot bind to this site. Okay, so when the calcium bind, this is the calcium. So when the calcium bind, there's a conformation change takes place. Again, I'm drawing. You can see that there's a change in the position, the conformation change again. Now this is C, this is T, and this is I. This is tropomyosin, this is troponin. Okay, and now this side, when the calcium bind, this side get exposed. The myosin binding site on the actin get exposed. Now the globular head of the myosin get attached to this side. Okay, so calcium play a very important role in bind to the troponin to remove the masking of the active site on the actin for myosin. So the correct option is 1. Okay. Second says that it activates the myosin ATPase by binding to it. No. Detach the myosin head from the actin filament. No, that is the role of ATP, detachment. Prevent the formation of bond between the myosin cross bridge and the actin filament. No. I hope it is clear to everyone now. Now moving next. Smooth muscles are. Please remember the feature of the cardiac muscles, smooth muscles, and the skeletal muscles. These are very important. Smooth muscles are, in option 1 says involuntary cylindrical striated, voluntary spindle shaped, uninucleated. Third says involuntary fusiform non-striated. Fourth says voluntary multinucleate and cylindrical. What do you think? What will be the correct option? It's very easy. Very good. So smooth muscles are also known as visceral muscles, right? They are involuntary muscles. So they are involuntary, yes. They're cylindrical, no. They're not cylindrical, they're fusiform. They're striated, no, they're non-striated. Very good, MS Gauri. Now, 2 says voluntary. No, they are involuntary. Spindle shape, yes, fusiform is a spindle shape. They're uninucleated, yes, they are uninucleated, but they are not voluntary, they're involuntary. Option 3 says involuntary, yes, they are involuntary. They're fusiform, yes, they are fusiform. They are non-striated, yes, they are non-striated. So correct option is 3. 4 says they are voluntary, no, they are involuntary. Multinucleate, no, they are uninucleate. Cylindrical, no, they are fusiform. I hope it is clear to everyone why the correct option is option number 3. Okay, now it's 1. We are winding up the class. I hope this lecture was informative for you. Thank you all for your participation and see you in the next class. Thank you and have a nice day.